inhibited because the stop process ended before the go process. And in fact, at this threshold, we have we can compute the stop signal reaction time. It's the difference between well, this threshold and the stop signal delay. That's our indirect way to compute this not overt behavior that is the process of computation of inhibition. So let's start and talk about Parkinson's disease. Throughout history, there are a lot of descriptions of this shaky palsy, starting from ancient Greece. But it's only James Parkinson that systematically described all cardinal features in 1817. And these features are tremor, bradykinesia, stiffness, and postural instability. Main reason of these symptoms is a loss of dopaminergic neurons in the substantia nigra pars compacta, specific nucleus in specific nuclei in our brain. And as you can see, this is positron emission tomography, or PET, and with a radioactive marker, we can mark the presence of dopaminergic neuron. On the left, you have the normal conditions of controls, and as you can see, the density of dopaminergic neurons is very high. It's represented in red. On the right, you have Parkinson patients, and the density is much, much lower because there is an active loss of dopaminergic neurons. But there are still other manifestations of PD, Parkinson's disease, that are not motor. So we have depression, dementia, sleep disorders, gastrointestinal disturbances, restless leg syndromes, pain, and postural hypertension, and urinary problems. But another non-motor manifestation is, in fact, that inhibitory control is impaired in Parkinson's disease. And here I've written proactive versus reactive with a question mark, because we don't really know how specifically proactive and reactive inhibition is affected in Parkinson's disease. So this is an active field of study. So how do we study Parkinson's disease? Here are some results from our lab. I will start with two studies. I will present them together because the first study tackles the reactive inhibition and the second the proactive inhibition. So please deactivate. Yes, thank you. So one influential theory states that inhibition of movements relies on a right lateralized network. So in other words, this scientist states that the right side of our brain implements inhibition. And we will use Parkinson's disease as a model to demonstrate that's effectively not what's happening. So this network is in the right hemisphere, and it's the inferior frontal gyrus, the pre-supplementary motor area, the subthalamic nuclei. So it's not important, the names are not important, but I will show you where they are located. And so Aaron et al. In 2007 suggested that these prefrontal areas are needed to inhibit your movement, and the right subthalamic nuclei are fundamental in this activity. But other evidence suggests that there is a larger network involved in this suppression of hand movements. So here in pink, you can see where the subthalamic nucleus is located. That's a frontal section. So the inferior frontal gyrus is, I show you here on the right. It's on the cortex, so it's easier to to understand where it is. It's more or less where your ears are located on your brain. And the pre-supplementary motor area in blue here are inside the medial part of the brain. So if you take the two hemispheres of your brain apart, it's on the inside. To tackle our question of right lateralized network or not right lateralized network for inhibition, we used deep brain stimulation. In fact, deep brain stimulation is an alternative way to alleviate symptoms of Parkinson's disease whenever dopaminergic treatment is no longer effective. And what happens? An electrode is implanted in the patient, as you can see here, and there are mainly three parts of the brain that are stimulated with this electrode: the globus pallidus, the pedunculopontine nuclei, and the subthalamic nucleus. And we will focus on the subthalamic nucleus because these patients that have an electrode on the subthalamic nucleus, or both nuclei, on the left and the right nuclei, well, they are perfect model to study our question, because the subthalamic nucleus is one of these key three areas that we want to assess for inhibitory control function. So our hypothesis is very simple. Given that we have an impairment of inhibitory control in Parkinson's disease, and given that we have a bilateral deep brain stimulation in Parkinson's disease patients, even if they are very rare, but there are still enough patients to do some research, then given that we can selectively activate and deactivate these stimulators during the administration of the stop signal tags that I explained to you before, well, then we can evaluate the role of the subthalamic nuclei in inhibition and answer the question, is it right lateralized or not? Here are the two papers, and let's see what we did. We took 10 DBS patients, so patients with Parkinson's and deep brain stimulation. So we tested them without dopaminergic treatment, without this treatment for 24 hours. So without, for 24 hours, they did not take any kind of dopaminergic treatment. So any effect that we see on inhibition is given by the stimulation of these electrodes that stimulate the subthalamic nuclei. Here, a brief overview of our 10 patients. Of course, our behavioral parameter is the stop signal reaction time that we compute as I explained before. And here are the results. As you can see, the controls on the left have a stop signal reaction time that is very comparable with the DBS patients that have an active electrode. So both electrodes are active. As you can see, there is no difference in reaction times. But what happens if we deactivate only the right electrode? Stop signal reaction times become much longer, meaning that patients without the active right electrode have a lot more difficulties to inhibit because they take a longer time to implement their inhibition. And this demonstrates that initially that maybe the right subthalamic nucleus is in fact the most important part of this network for inhibition. But what happens if we deactivate only the left? Well, the same. If we deactivate only the left and keep the right active, we have the same result. The patients have the same difficulties to inhibit the stop signal reaction time. It's much more longer, and it's not important if you have only the right or the left electrode active. So it does not seem that there is a lateralization of this network. In fact, if we deactivate both electrodes on the right and on the left, we have exactly the same results. Inhibition is impaired, or better, reactive inhibition is impaired. But what happens to the proactive inhibition? So, as I told you before, we see the differences in reaction times and movement times between the stop signal task and the go-only task. The go-only task is this simple task where you only move to the right and you have no stop signal. As you can see, these are cumulative probability distributions. It's not so complex as you can imagine. Just to remember that if the distribution is on the left, then on average the reaction time is shorter. If you have the distribution on the right, then on average the distribution is longer, so it takes longer. In red, you see the reaction time of the go only, and of course they are much faster. I'm showing you the results of our healthy controls. And on the right, you see the reaction time of no-stop trials. So the same movement, but in the no-stop trials, the controls know that there could be a stop signal. So the reaction times becomes longer because they wait for the stop signal. They proactively wait for a stop signal. And here are the movement times on the dotted lines. As you can see, the movement time of the go signal, of the go task is more on the right than the movement time of the no stop signal. So even if the difference is not very great, it's consistent throughout all the distribution. So it's extremely significant. And that's exactly what happens. We call this the context effect. So given a specific context, in our case, the presence or absence of stop signals, even if you do the same movement, your reaction time and movement times completely change in length. Here are our patients with an active, with both electrode active. You can see on the reaction time, it's perfectly the same, more or less, than controls. The movement times are in general a little bit longer, but just because they are patient, they have a little bit more difficulties to move. But the important thing is that you have a healthy context effect. So proactive inhibition is working when you activate your electrodes. So what happens? You see the movement times of no-stop trials, the dotted black line that is on the left, so it's shorter, and the red dotted line on the right, it's a little bit longer. So we have a perfect context effect when we activate our electrodes. And this is when we deactivate both electrodes. So as you can see, the reaction times remains the same, but it's not important. The important part are the movement times. And if you compare it to the on or to the controls, the movement times, the dotted lines, are inverted. So we have longer no-stop trials and shorter go trials. This means we have a so-called inverse effect. We don't see this context effect on movement times, meaning then without electrodes, we have unimpaired proactive inhibition. So let's see. If we have another approach to tackle proactive inhibition. We see how single subjects behave. And what we do, we try to see if they have a context effect, as I told you before, and or a reversed effect. And it's the same as before, but we count how many subjects have this context effect and how many do have an inverse effect. And here you can see on the left controls, the majority of context effect. Sorry for the helicopter, and maybe you're hearing it. Okay. Okay. So the controls on the left have mainly a context effect. So the subjects have mainly a context effect. In fact, only one has a reverse context effect in movement times. And when you activate both electrodes, you see that the vast majority of patients have a healthy context effect. So proactive inhibition is working. But if you deactivate both electrodes, well, then this context effect disappears, and most of them has a reversed effect. A brief discussion of these first two papers. Only if you activate both electrodes, then you have, and that meaning both subthalamic nuclei are, then you have an improvement of reactive inhibition. And that's important because this kind of surgical procedure to implant these electrodes, it's very costly. And what happens is that most of the times the doctors decide to implant only one electrode on the left or on the right side. But here we demonstrate that for a healthy control, inhibitory control, we need both electrode active. So this data helps the doctors to decide that maybe it's better to implant both electrodes and not only on the right or the left side, because the progression of the disease cannot be hold. So we need to think on the long term, and this data helps the clinicians to decide to maybe implant both electrodes. And then another take-home message is that we have no evidence for a right lateralized inhibitory network. In fact, if we deactivate only one side, only one electrode, we have the same impairment as deactivating both electrodes. And finally, bilateral stimulation of the subthalamic nuclei restores, to some degree, effectively the proactive inhibition. And that's very interesting data. The next paper that I want to present, the next work that I want to show you is this paper. And here the hypothesis is more or less it's similar, because it has been shown that the subthalamic nucleus stimulated by this electrode, this deep brain stimulation electrodes, modulates the cortical plasticity in the primary motor cortex. So it could be that what we have seen before is just an effect of cortical plasticity. So in other words, chronic bilateral stimulation of the subthalamic nuclei could have induced some form of brain plasticity that has altered inhibitory control. So if you want to see what happens only with both electrodes active, well, then we see an improvement of inhibition. But we can use a rare model that is unilateral subthalamic deep brain stimulation patients. These are patients that have only one electrode installed and not 2 electrodes as before. So if the role of the right subthalamic nucleus was masked in previous study by a bilateral deep brain stimulation activity, so cortical plasticity, now by using only the right unilateral subthalamic DBS patients, we could exhibit a better inhibitory control, because there should not be any cortical plasticity between the 2 hemispheres. Again, we selected 10 right and 10 left patients. So 10 patients had a left implant and 10 had a right implant. And then we used 22 healthy controls. This time they were on dopaminergic treatment. So overview of our patients, and here are the results. The control is in green, and as you can see, the stop-signaling reaction time is very, it's relatively short, about 220 milliseconds. And here we have the left deep brain stimulation on the left, and patient with the left deep brain stimulation off on the other side. So in yellow, we have patient with only the left deep brain stimulation. And it is not important if the left DBS is active or it's off. There is a clear inhibition, clear deficit in reactive inhibition. Stop-signaling reaction times are much longer with respect to the controls. And here are the right patients with the right implant. And even here, you can see there is no clear difference. All these papers, all these results are not significant. We computed Bayesian factors to compute the extent of the alternative hypothesis, and it clearly shows that there is no difference. It's not important if you have the DBS on the right side or on the left. If it's on or off, it does not work. There is no improvement in reactive inhibition. But here is some brief data about proactive inhibition. More or less, you can see that they behave similarly on reaction time as expected, but it's a little bit more complex to understand that. But in the controls, you can see that the no-stop trials are shorter with respect to the go-only trials, as it should be. But you don't have this kind of clear differences in any combination of our patients. If you use the single subject approach as before, you can see that the controls on the right, the vast majority of them have a healthy context effect. So proactive inhibition is working. It's shown, the context effect is shown with the right bar, but there is no clear evidence of patients having any kind of better proactive inhibition. More or less, if you sum no context effect with the inverse effect, it's roughly the same as the context. So it's not above chance level. Brief discussion of this paper. So the unilateral DBS of the subthalamic nuclei does not improve reactive inhibition or proactive inhibition. And these results are clearly in line with our 2 previous papers that we published. And the null results have been interpreted with, must be interpreted with caution. And as I told you before, to give some effect to our null results, we used Bayesian factors that confirmed that there is no difference in the stop signal reaction time in any combination of patients and on-off state. So there is no evidence for a right lateralized inhibitory network, even with this approach. And let's go forward. I think that's the last paper that I will present you. So in this paper, we tackled we tackled always the problem of the lateralization of the inhibitory network, but we used another approach. So in Parkinson's disease, it happens that you start usually to have your symptoms even on the right side or on the left side of your body. For example, it could happen that you start to have your symptoms with your right hand or right leg. That's because your left substantia nigra pars compacta, your left nucleus where your dopaminergic neurons are, they start to die before on the left side, so you see your symptoms on the right side of your body. And it can happen on the other side, and the left side of your body is starts to have some problems. That's because your right dopamine neurons are starting to die. So Parkinson's starts always on one side or on the other side, and we can use patients that were diagnosed with Parkinson's disease and use this information. And effectively, left dominant PD patients should be more impaired because the right substantia nigra is impaired. And as you can remember, the right side should be the right the part of the network that uses inhibitory control, that serves inhibitory control. Whereas the right dominant patients that start with symptoms on the right side, they should be better in inhibition if this theory of right lateralization is correct. So we collected 20 patients that have left symptoms and 20 patients that have right symptoms and 20 healthy controls. We choose only patients with a pronounced symptom asymmetry, and they did our stop signal reaction time as before, and they were on therapy. Here is a brief summary of our patients, and here are the results. As for reactive inhibition, again, there is no difference between right or left patients. They both are significantly more impaired than controls. As you can see, they have longer reaction times than controls, significantly longer reaction time. But between the two patient types, there is no difference. We checked even the difference in arm, because if you use the right arm, you are effectively using the left side of your brain and vice versa. So this effect could be masked by the fact that you're using only the right hand to do your stop signal task. So we administered the same task to 15 left and 15 right patients. Both we administered the stop signal task using the right hand, and then again using the left hand. So it was a little bit more complex procedure, but as you can see, there is no significant effect, and more or less they all hover around the 260 milliseconds stop signal reaction time. So there is no effect of hand that says to us that there is no difference, even if you include the factor of arm. And what's about the proactive inhibition? Again, we look on the movement times, and if you see the median, that's the right here, the right the red line that marks the fact that 50% of participants have a lower value and 50% have a greater value. As you can see, in controls, you have for the stop trials a shorter movement times than the go-only trials, as expected. This means there is a correct, healthy context effect. But here with the patients, there is a little difference in movement times generally between the two trial types. There is no difference in movement. The median is always on the same level. So again, there is no difference in proactive inhibition. And this is the second approach, by counting how many patients have and how many controls have a context effect or an inverse context effect. Again, the controls here have, the majority of them have a healthy context effect, but patients, it's not important if they are right or left patients, they both have the same amount of context and no context effect, meaning this effect is not above chance level. Finally, we checked the effect of the right and the left hand, but there is no significant difference of the effect of hand. So, brief discussion. The results suggest that in reactive inhibition, so what we measure with the stop signal reaction time, there is an equally impaired, they are equally impaired in the left and right Parkinson patients. The pattern of impairment in proactive inhibition is very similar in the two groups, so we cannot say there is difference in between patient type. So there is no evidence for right lateralized inhibitory network. Again, okay, now this is the last. Sorry, this is the last paper that I will present, and it's the continuation of this the paper that I just told you, but we made more or less the same experiments using early stage Parkinson's disease patients. So in the last study, we used patients in the Hoehn-Yahr stage 2 and 3, meaning patients that are advanced in their pathology. But even if they show asymmetrical symptoms, both hemispheres could be affected just by the fact that they are on a very late stage of the disease, and so it could be that what we see, this absence of difference between right and left, is just because they are very affected patients with a long history of this disease. But if we use early patients that are diagnosed and are in an early stage of this pathology, well, then this argument can be overthrown. I mean, if there are in the early stage, we can effectively evaluate the reactive and proactive control and check if the left or the right Parkinson's patients have some kind of difference. So we can again check if the right lateralized network is involved or a more dispersed network is involved. So we collected data from 17 early left and 17 early right Parkinson patients, and in both groups 10 were drug naive, so they just were diagnosed with Parkinson's, and then they did not start any kind of medication. So we just had the luck to test them before they started any kind of medication. And other 7 in both groups had stable treatment with low doses of dopaminergic treatment because they just are in the early stage of Parkinson's. Patients performed again with the right arm and with the left arm, so they repeated the stop signal task 2 times, one with the right and then another time with the left arm. And we collected data of 24 healthy subjects. Here is an overview of our patients and controls. And here are the results, more or less the same. It does not matter if you are in the early stage or not. If you're right or left Parkinson patient, your stop signal reaction time is longer, significantly longer, with respect to controls. Here again, some results about proactive inhibition. These are our patients. The dotted lines are again the movement times that are the most important part of proactive inhibition. And as you can see, there is they take patients with the right side take a little bit longer to make the movements as expected, but they show a correct and healthy context effect. So the movement times of no-stop trials shorter, so on the left, than the movement times of go-only trials on the right, and it's highly significant. Whereas the left patients show more or less the same effect. As before, check out the dotted lines. They are on the correct side for the correct context effect. So this is the average of the population. Of course, controls are faster than patients and are shown in green. There is difference again overall about movement times, but there is also a difference in context effect. In all 3 groups, as you can see, no-stop trials are shorter than go-only trials, exactly as you would expect. So in this case, early Parkinson patients have an intact proactive inhibition, but not unimpaired reactive inhibition. This is the single subject approach. As you can see, early stage Parkinson's patients show the same pattern and have a majority of subjects with a healthy context effect. Finally, discussion. As I told you before, patients have an impaired reactive inhibition, so their stop signal reaction time is longer, but the proactive inhibition is not impaired, and that's an important finding. So there is no difference between drug-naive and treated patients. And inhibition does not change between the arm employed. I don't have data, but there is no difference in arm as before. So no evidence again, and I think I can convince you that our inhibitory network is not right lateralized. So this is given the fact that we did not find any difference in patient groups. And that's all from me now. Thank you very much for your attention. I hope it was not too difficult to understand, and wish you good luck.
Thank you very much for the presentation.
If there are questions, I'm happy to answer your questions.
Anybody have a question? You can raise your hand or send your question in chat.
Okay. No questions?
Just Noor asked if may she have your email.
Okay, well,
If there is no questions, please fill the survey with the link in chat.
sorry?
For the participants,
For the
there is
participants,
a
sorry.
way to fill.
Well, if there are no questions, I will.
They just thank you for the presentation.
And thank you for your participation and attention. Thank you very much.
Thank you for the presentation.
Thank you. Bye.
Please fill the survey before you left. Thank you, everyone.
Excuse me, the survey link not valid, not invalid.
Okay, I will send another one. Thank you.
Okay, okay, thank you.
Check this, please.
Okay, thank you. Now valid. Thank you.
Welcome.
طلع هذه لا سبحان الله طلع روحك. يعطيك العافية.
الله يعافيكي.
بس أنا نور، أنا كنت بس بدي أسأل هل هو سمع إنه طلبت الإيميل تبعه أو ما؟
والله أنا حكيت له.
شكله ما انتبه.
ممكن نبعث له إيميل خبره إذا في مجال نبعث لك الإيميل تاعه.
موجود عندكم يعني أو عندك؟
بعتي لي الإيميل تاعك بالشات.
يلا هلا ببعته. هاي اندعت.
تمام، إن شاء الله بنخبره وبنتواصل معك.
شكرا لك.